So this is the first part of chapter 6. We're going to talk about adaptive immunity. And the reason I say it's the first part because last spring we were right in the middle of this chapter when we went into lockdown. So you're actually going to get a first part of chapter 6 and a second part of chapter 6. So let's take a look at chapter 6. So we've already talked about the types of immunity where we said that we had the innate immunity and adaptive immunity. What we'll see in adaptive immunity are two types. We have the humoral immunity, which is under control of the B lymphocytes or B cells, and this is going to be involved in the production of our antibody proteins to antigens, and it is the main defense against bacteria or their toxins versus cell-mediated immunity, which is under control of the T lymphocytes or T cells. This is the formation of a population of lymphocytes that will attack and destroy, quote, foreign material, and really I will say non-self material. This is the main defenses against most viruses, some of the fungi, parasites, and some bacteria as well. Cell-mediated immunity is also the mechanism by which the body will reject transplanted organs or tissues. And it's a means of eliminating abnormal cells that arise spontaneously in cell division, and this includes cancer cells. So let's look at a comparison between the two. Humoral immunity, again, is under control of the B lymphocyte. It responds to bacteria, bacterial toxins, and some viruses. When activated, B cells form memory cells and plasma cells. The plasma cells are the cells that actually produce antibodies to the foreign antigens. Whereas cell-mediated immunity under control of the T cell, these respond to cancer cells, virally infected cells, single-cell fungi, parasites, and foreign cells in organ transplants. When activated, the T cells also will differentiate into memory cells, but they also will form cytotoxic cells, suppressor or regulatory cells, and helper cells. The cytotoxic T cells are the ones that will attach to the antigen directly and kill whatever the antigen is attached to. The antigen must first be processed and displayed on the cell membrane of what's known as the antigen-presenting cell before the adaptive response can be set in motion. Lymphocytes interact with the antigen they are programmed then to recognize from these antigen-presenting cells. When they are appropriately stimulated, the B lymphocytes proliferate into memory cells and plasma cells that then will produce antibodies to this specific antigen. T lymphocytes will proliferate into a diverse population of cells that regulate the immune response and generate the cell-mediated reaction to eliminate the antigen and whatever it is attached to. The initial contact with a foreign antigen is followed actually by a lag phase of about a week or so. Basically, we usually say that the lag phase in this first contact is 5 to 10 days. And this is before an immune system or immune response can actually be demonstrated easily. After the body's immune mechanisms have reacted to the foreign antigen, some of both the B and T cells will retain the memory of that antigen that induced the sensitization, so that when we then see this for a second or subsequent time, we'll have a faster response. The memory is actually passed to succeeding generations of these B and T memory cells so that we actually develop millions of these memory cells to this specific antigen. So later contact with that same antigen provokes a stronger and, more importantly, a faster proliferation of sensitized lymphocytes and antibody-forming plasma cells. So in our primary response, what we're going to see is here's our initial antigen exposure. And oftentimes what we're going to actually be looking at is the level of antibodies in the bloodstream. And so what we'll see is that we have this 5 to 10 day lag period where we do not see an increase of antibody levels in the bloodstream. It isn't until that lag time is complete that we start to see a rise in this primary immune response, but we actually will use up those antibodies fairly rapidly in fighting whatever this antigen is. But in our secondary response, our second antigen exposure, our lag time will only be about 1 to 2 days before we see a significant rise of antibodies and a much slower then decline because we've just made so many extras at this point. Now, if we see this antigen a subsequent time out here somewhere, then we're going to basically see no lag time because our antibodies from our secondary response here actually hasn't gotten down to base level, and so they can respond immediately, but we will still see then another higher increase in antibody level and usually a much slower decline over time. And so this would be a subsequent secondary immune response. The reason why we have a stronger and larger effect is because the body's like saying, well, okay, so we saw it once, we saw it a second time, now we're seeing it a third time. Why don't we just keep our antibody levels up really high so that as soon as that antigen enters the body, the antibodies can bind to it and we can start getting rid of it before it could potentially cause illness. This is also why we typically give boosters on vaccinations. We hope that the first exposure of the vaccine will produce the primary immune response and memory, and secondary and subsequent exposures to boosters then will produce even a stronger response. So entry of a foreign antigen into the body triggers a chain of events. First of all, we have to recognize that it is foreign to the body and distinguish that from self. We'll see a proliferation of lymphocytes that are programmed then to respond to this antigen from a large group of cloned cells. What we're going to see is an antigen presenting cell will present an antigen to a T helper lymphocyte that can receive it. That now sensitized T lymphocyte will undergo clonal expansion. It will begin to undergo mitosis, and all the subsequent generations of that sensitized T cell are also sensitized to that antigen without directly having to receive that antigen from an APC. Then that T helper cell that I call the general of the immune response will send out chemical messengers to all the other cell populations, and this will eventually result in destruction of the foreign antigen either by antibodies produced from the plasma cell of the B cell response or from cytotoxic T cells of the T cell response. So we have to talk about something called the major histocompatibility antigen complex, because these are going to be involved in the displaying of a processed antigen on our antigen presenting cells. The major histocompatibility complex presents processed antigens to responding cells, primarily the T helper and cytotoxic T cells of the immune response. They're active in generating an immune response. They're coded for by the HLL gene complex that's located on chromosome pair number pair 6, and they control the immune response by regulating both T and B cell proliferation. They influence resistance to infection and tumors, and they also will increase likelihood of acquiring an autoimmune diseases because some of these actually are what we call our self-markers or self-antigens. So the major histocompatibility complex proteins, there are basically two major classes that we're going to discuss anyway. And what I want you to know is what membrane of what cell are they found on, what's their primary function, is it recognized by which receptor, and recognition receptor is found on what cell. So, the class 1 major histocompatibility antigen complex proteins are found on all nucleated body cells. This includes every cell in the body except the erythrocyte as a mature cell. They have their own major histocompatibility complex proteins, if you will, in the ABO and RH blood groupings. The function of class 1 is to present endogenous or self-antigens. So these are our true self markers found on all our cells. They are recognized by the cluster of differentiation 8 receptor, or what's known as the CD8 plus receptor, and that CD8 receptor is found on the cell membrane of cytotoxic T lymphocytes. Class 2s are found on specialized immune cells, including all of the lymphocytes and the professional antigen-producing cells. And this includes macrophages, dendritic cells, which are specialized macrophages, histiocytes, which are macrophages that are found in specific organs, and the B lymphocytes themselves. The class 2 present exogenous antigens, so those that actually arise from outside the body. The class 2s are recognized by the CD4 plus receptor that is found on the membrane of the helper T cell. So, the role of lymphocytes in the adaptive immunity. Again, we're going to have two groups, main groups of lymphocytes, the T and B lymphocytes. Now, I can't look at a lymphocyte in the bloodstream and tell you whether or not it's a B cell or a T cell. What I can say is it's most likely a T lymphocyte because we typically have more T cells circulating at any given time than B cells. The macrophage and the other professional antigen presenting cells, and so this is our big macrophage over here, is what's going to actually present the antigens to these lymphocytes. So, both the T and B lymphocytes are made in the bone marrow. So they both come from hemopoietic stem cells that develop into the two stem lines of the B and T lymphocytes. So both of them are made in the red bone marrow. However, each of them is going to be endowed with a specific receptor found on their cell membranes. And this is going to help in their ability to do their jobs. And so I say that they basically receive their specialized training. Well, where does that specialized training actually occur? For the B lymphocyte, that actually occurs in the bone marrow, hence the B cell designation. But the T lymphocytes actually are released from the red bone marrow into the bloodstream where they travel as immature cells to primarily the thymus gland located in the anterior thoracic cavity. Here they will receive their T cell receptors, and they then will leave the thymus gland. The B cells will leave the bone marrow and go into circulation, and oftentimes both will actually then take up residency in the secondary lymphoid tissue, which includes lymph nodes, the spleen, and any other lymphoid tissue that we find out there. So we can say that the red bone marrow and the thymus gland are what we call primary lymphoid tissues, and everything else is a secondary lymphoid tissue. Once they actually take up residency in these secondary lymphoid tissues, they are not yet activated, but they are now primed in order to receive the information from the antigen presenting cells or from chemical mediators. T lymphocytes are unable to respond to a foreign antigen until one of these APCs has phagocytized the antigen, digested it in the phagolysosome, and displayed it on its cell membrane on one of the classes of MHC proteins. Since this would be a foreign antigen in this case, it's going to be displayed on an MHC class 2 MHCs. The helper T lymphocyte responds to the T cell receptor by its T cell receptor and the CD4 receptor to the MHC class II proteins with that presented antigen. This will now activate or sensitize this T helper cell to that specific antigen. This T helper cell will now undergo clonal expansion, and every subsequent generation of that sensitized T cell will also be sensitized to this specific antigen. Cytotoxic T lymphocytes will respond by the T cell receptor and the CD8 receptor to the MHC class I proteins presenting endogenous antigens, and they actually will then become activated to release a group of chemicals known as perforins and granzymes that will destroy whatever that antigen-bearing cell is. Helper T cells, and there are actually several subpopulations of helper T cells, but these are the effector T lymphocytes that regulate the other cells of the immune response by releasing a number of cytokines, or again, cytokine simply means cell mover. They are chemical mediators of this response. So in HIV infections, the virus actually attacks and destroys the T helper cells. So people with HIV don't die of AIDS, they die of other infections that the immune system can no longer bring under control because they've lost the T helper cell, which is really the key for the adaptive immunity to start. Cytotoxic T cells, once they've been sensitized, will seek out and destroy abnormal body cells. This will include cancer cells, or virally infected cells, or any other abnormal cell. Regulator T lymphocytes. This is a subpopulation that actually comes from the T helper cells that will help regulate the immune system by establishing a balance between promoting and inhibiting the immune response. Let's say that we've had an exogenous antigen, we've had the immune response, we've gotten rid of the antigen-bearing substance. Do we now need to downregulate these sensitized cells so that, in fact, they don't start attacking our own self cells? And that is very, very true. In fact, one of the things that we oftentimes think now with autoimmunity is that part of that autoimmune disease may be due to the loss of function of these regulatory T lymphocytes. So this just shows a macrophage, let's say as an APC here, it's got an exogenous antigen on the class II. It is presenting that antigen to the T cell receptor along with the CD4 receptor will be involved with this, and that recognition will sensitize that T cell to this specific antigen. If this is an endogenous antigen, then we'll put it on a class I and present it to the T cell receptor on the CD8 receptor of the cytotoxic T cell. This cell is now sensitized to recognize that antigen anywhere out in the body, to attach to that cell, and to secrete perforins and granzymes that will destroy the cell that the cytotoxic T cell is attached to. Two types of effector T lymphocytes are restricted in their ability to respond to processed antigens complexed and presented with the MHC proteins. And these include the cytotoxic T cells, also known as the CD8 cells. Again, these can only respond to antigens complexed with the MHC class I displayed on infected host cells, so self cells that are abnormal. Helper T cells, also known as the CD4 cells, can respond only to processed antigens displayed on macrophages or related APCs, along with the MHC class II proteins. Now, where do the B lymphocytes fit into all of this? Well, the B lymphocytes also are given a specialized B cell receptor. And we now know that this is actually the IgD class antibody, which the constant region will be embedded into the cell membrane of the B cell. These IgD class antibodies then will function as antigen receptors, and they can bind an entire antigen molecule to their receptor. This will then cause movement of this antigen into the cell, and it is then processed into fragments, just like any other antigen presenting cell. And that's why B lymphocytes can actually be and are included in the professional antigen presenting cell group. The fragments then will be displayed on the class 2 MHC protein. Now, B lymphocytes can either be activated directly by the antigen, or they can be actually activated by lymphokines, one of the cytokines that T helper cells release to actually begin to produce antibodies. So the antibodies are these globular proteins produced by the activated or effector B lymphocytes called plasma cells. The antibody protein is made up of 4 polypeptide chains, 2 shorter ones known as the light chains, and 2 longer or heavy chains, and these are all held together by disulfide bonds, and they form kind of this Y-shaped structure. Each FAB region, or the highly variable region right here on the arms, is the only part of the antibody molecule that can react with the specific antigen. The reason for this is the amino acid sequence in the upper arms here of the FAB region, and this stands for antigen binding region, will fit only one antigen in its makeup. So this is why these areas are variable in their amino acid content. The FC region stands for constant or crystallizable fraction, and in these regions, the amino acid sequence does not vary very much within a class of antibody. So I can look at this space filling model here, and I know that there are some carbohydrates right here on the FC region, and because of their location, I can tell you that this is an IgG class antibody. Antibodies are also called immunoglobulins or gamma globulins. And in fact, in your bloodstream, you have the big group of proteins called albumins. The second largest group of proteins is called the globulin proteins. In the globulin proteins circulating in your plasma are the alpha, beta, and gamma globulins. So these would be your circulating antibodies. There are 5 classes of antibodies, and they each do very specific things, even though there's overlap amongst the classes. The I little g stands for immunoglobulins, and the capital letter stands for the specific class. IgG class antibodies exist in the blood as a monomer, and this little figure right here is representing carbohydrates attached to the FC region. These are the longest lasting in the blood, and they oftentimes are made in large numbers, especially in secondary immune responses. They also have the unique characteristic of being able to cross from maternal blood to fetal blood across the placental membrane. This sometimes can be a good thing, and it sometimes can be a bad thing. Can you think of a time it might be good, and can you think of a time that it might be bad? So they're smaller in the fact that they exist as a monomer or a single antibody protein. They're the principal antibody molecule in response to the majority of infectious agents like bacteria. And they are the only class that crosses the placental barrier to go into the fetal bloodstream. Now the IgM class, this is the largest in size of our antibodies. There are actually 5 antibodies, and so this exists as what we call a pentamer, and they're held together by something known as a J chain. Each one of these antigen binding fractions could potentially bind to an antigen, and so this has 10 docking sites for antigens, and we call that the valence. So IgM class has a valence of 10, while IgG only has a valence of 2. This is a large antibody. It's a macromolecule, a pentamer, and it will always be the first antibody class that is produced in an antibody response. So in that primary response, the first class we'll see is IgM, but they are rapidly used up because they have so many areas to bind up antigen. IgA is a monomer in blood, but we find very little IgA actually circulating in the blood. Where IgA actually migrates to is in the body secretions, where it exists as a dimer held together by this J chain. So it has a valence of 4. This is important that it's in the body secretions because now it can actually bind to antigens before they may even get into the body. So produced by the plasma cells located in respiratory and GI system primarily, and they will then combine with harmful ingested or inhaled antigens, forming antigen-antibody complexes that cannot be absorbed into the body, preventing the antigens from actually getting in and inducing sensitization. IgA is found in sweat, tears, saliva, mucus, and breast milk. And so this is another important class of antibodies because it is actually going to help with the newborn's immune response. IgD exists as a monomer in blood, but very, very little is found in the blood. Usually less than 0.02% of your circulating antibodies will be of the IgD class. When these are made, they are actually inserted by the Fc region into the cell membrane of maturing B lymphocytes. So this is actually the B cell receptor. So present in very minute quantities actually in the bloodstream. And then IgE also is found in very small amounts in the blood because this one also will be attached to a cell by the Fc region, and this is going to actually be inserted into the surface of mast cells and basophils. IgE is oftentimes involved in allergic reactions or hypersensitivity reactions, and one of the reasons is because the mast cell has basically been sensitized to an antigen or an allergen in this case by the IgE present, and this, when the antigen binds to the antibody on the surface of the mast cell, it causes the mast cell to undergo degranulation and cause the inflammatory response. So concentration of IgE is oftentimes increased in allergic individuals on the surface of their mast cells. IgE is also involved in parasitic infestations. Now, I mentioned the complement system in inflammation. Well, the complement system is a blood plasma cascade that will function with the immune system to destroy or inactivate types of foreign antigens. It's activated by the antigen-antibody complex in what's known as the classical pathway. The alternative pathway is actually due to a small protein called lectin that binds to some small proteins found in the plasma that will actually activate the complement cascade, or the mannose binding lectin pathway. And this is simply another pathway. And in this case, it actually has to have mannose, which oftentimes is a component of especially gram-positive cell walls. Complement proteins that are produced by the complement cascade then will interact to accomplish several different functions. One is they can mediate and actually enhance inflammation. They will coat the surface of invading bacteria, causing something known as opsonization. Opsonization is basically highlighting something for phagocytosis. and they can generate something known as the membrane attack complex. And this is actually a series of these complement proteins that act together to punch holes in foreign cell membranes. So the classical pathway of the complement is again antigen antibody dependent, and this will form on pathogen surfaces, and this will actually do what we call fix complement or start this side of the cascade. The alternate pathway, C3 protein dissociates again by the presence of lectin and some other little proteins into its active form of C3A and C3B. And the lectin mannose pathway will actually bind to the mannose on the protein surfaces of the especially gram-positive bacteria. No matter which way we activate the cascade, it all comes down here to the activation of C3 to the active forms of C3A, C3B. And these both then have different responses. C3A can bind to basophils and mast cells and cause the release of the inflammatory mediators, actually causing an increase of inflammation. And C3A also can stimulate neutrophil and macrophage activity, increasing phagocytosis, which increases inflammation. C3b has a number of different things. It can bind to antigen antibody complexes to red blood cells, and the red blood cells then will transport those antigen antibody complexes to the liver and spleen, where phagocytes will actually remove them, and so it functions in immune clearance or increase of phagocytosis. It can coat bacteria for phagocytosis by opsonization, and it will split complement 5 into its active form, and along with C6, 7, 8, and 9, this will form the membrane attack complex that will result in cytolysis. So complement actually complements or helps with inflammation. So this last slide is a simplified version of the immune response. And you look at this and you go, it's simplified, and yes, it is simplified. Hopefully we get to spend some time with this in lecture together to go through this so that I can kind of explain it. But I'll just mention here that this right here, to phagocytosis and really kind of coming up through complement, is inflammation. So this right here is inflammation. The rest of this, from phagocytosis through the T and B cell side, is going to be the adaptive response. And you can see that it is actually cyclic in nature. The key to this whole thing is actually right here. Both inflammation and the adaptive response increase phagocytosis. In order to heal an area of tissue injury, we must first clean it up. And so I use this term. In order to heal it up, we must first clean it up. So, inflammation, we get immediate vasoconstriction, we get bleeding. The bleeding will be taken care of through the platelet plug and the formation of the fibrin clot through blood coagulation. The kinin system will be activated at the same time as blood coagulation through factor 12. That produces bradykinin, which enhances pain in the inflammatory response. The mast cells degranulate almost immediately. This produces histamine, heparin, which is actually a short-acting anticoagulant that will help increase blood flow to the area, but then it's gone. Okay. Serotonin, prostaglandins, leukotrienes, all of those are going to cause vasodilation secondarily. That will increase blood flow to this area. We will then see edema and other signs of inflammation. Some of these are also chemotaxic, and so we'll see increased leukocytes into this area of injury, with the neutrophils being primarily in the early stages, macrophages coming in for the later stages. All of this will increase phagocytosis. Now, some of those phagocytes are the antigen-presenting cells. Antigen-presenting cells will phagocytize whatever this foreign substance or abnormal substance is and process it and present it to the T helper cells. The T helper cells will then undergo clonal expansion. Some of those T helper cells will become memory cells that will remember what this antigen is. Some will become the regulatory T lymphocytes that can have negative influences then on the other cell populations to help modify and regulate the response. The T cells will become activated to endogenous antigens. They can then actually attach to an antigen-bearing substance and release perforin and granzymes that will cause apostosis of that antigen-bearing cells. The T lymphocytes will release interleukins, lymphokines, and other cytokines. Some of the interleukins will cause systemic fever. That will cause an increase of phagocytosis. Some of the lymphokines interleukins will activate B cells to become activated plasma cells. These cells can also, or will also, actually become, or some will become memory cells. And I didn't put the arrow in because there was just too many arrows going on. The plasma cells will begin to produce antibodies to the specific antigen. The antibodies go out and bind to the antigens to form antigen-antibody complex. That alone will increase phagocytosis. The antigen-antibody complex will also activate complement through the classical pathway. That will cause chemotaxis, increasing phagocytosis. Production of opsonine proteins increases phagocytosis. As phagocytosis increases even more antigen-presenting cells will process more of this antigen and present it to more T helper cells, which will just continue the cycle. So I hope that we get to be able to talk about this actually in class together during lecture. But if we do not, I will actually give you some videos on this in a little bit more detail.